Hi friends, welcome to our channel Learn with Snake. In this video we are going to talk about a history of immunology. First I am going to explain about Thucydides. He described a plague in Athens. He wrote in 430 BC that only those who had recovered from the plague could nurse the sick because they would not contract the disease a second time. Although early society recognized the phenomenon of immunity, almost 2000 years passed before the concept was successfully converted into medically effective practice. Second, in 15th century, Chinese and Turks deliberately induced immunity against smallpox, which is fatal about 30% cases. So that dried crust derived from smallpox pustules were either inhaled or inserted into small cuts in the skin, and the technique is known as variolation, and positive effects were observed. Third is about Edward Jenner. He made a giant advance in the deliberate development of immunity against targeting smallpox. In 1798, the milkmaids who had contracted the mild disease cowpox were subsequently immune to the much more severe smallpox. Jenner reasoned that introducing fluid from a cowpox puzzle into people, that is, inoculating them, might protect them from smallpox. It nearly takes 100 years before this technique was applied to other diseases. Next major advance in immunology is the induction of immunity to cholera. So, Louis Pasteur has succeeded in growing the bacterium that causes foul cholera in culture and confirmed this by injecting old culture into the chicken, then developed fatal cholera. After some days, he injected the culture to some chickens. Unfortunately, the chicken became ill, but they recovered again. So again he grew a fresh culture of bacteria with the intention of injecting this lethal brew into some unexposed fresh chickens. But he injected the new culture into old and new chickens. Unexpectedly, only fresh chickens died and old chickens were alive. So Pasteur hypothesized and later showed that aging had weakened the virulence of the pathogen, and thus such a weakened or attenuated strain could be administered to provide immunity against the disease. He called this attenuated strain a vaccine in honor of Jenner's work with cowpox inoculation. Pasteur extended these findings to other diseases, demonstrating that it was possible to attenuate a pathogen and administer the attenuated strain as a vaccine. So, in 1881, Pasteur first vaccinated one group of sheep with anthrax bacteria that were attenuated by heat treatment. Then he vaccinated virulent culture of bacteria to vaccinated and unvaccinated sheep. As expected, vaccinated sheep were alive and unvaccinated were died. These experiments marked the beginning of the discipline of immunology. In 1885, Pasteur administered his first vaccine to human, a young boy who had been bitten repeatedly by rabid dog. He was inoculated with series of attenuated rabies virus preparations. Thank you. Thank you for watching this video. Please like, share and comment. Thank you.